In this section, I'm going to talk about some major types of fungi, and then I'm going to talk about some select fungal pathogens. So there's four major phyla of fungi: Zygomycota, Ascomycota, Basidiomycota, and Cryptococcus. Zygomycota, also known as the Zygomycetes, produce zygospores. They're usually black in color, and you can see them found on bread. This is a pretty common bread mold. It can be found on rotten fruit and vegetables. It can be found on the backs of flies, so flies can spread this mold around and spread spores around all over the place. And then it can be found growing on cow dung because it lives inside of the cow and it gets passed out into the feces, and then once it's on the ground it can grow and sprout up its spores and then replicate that way. There's only a few human pathogens that are Zygomycetes. Rhizopus has the potential to be one. Mucormycosis is another potential pathogen, and then there's select few others. Ascomycetes are known as the sac fungi. So Ascomycetes is a large group containing fungi with various lifestyles. There's all different types of Ascomycota, but in this group, these contain a majority of the clinically significant human pathogens. So most of the human pathogens are found in the group Ascomycota, and also baker's and brewer's yeast is found in Ascomycota. Basidiomycetes, or the Basidiomycota, contains familiar fungi such as puffballs and mushrooms. So those organisms have the ability to produce these fruiting bodies such as mushrooms. And chytrids, or in Cryptococcus. Cryptococcus contains spores that are flagellated. So flagellated means that they have a flagella. This allows the spores to swim in water sources. Now there's a reason why this frog is pictured here under the chytrids or Cryptococcus. Frogs have pores in their skin, and it allows them to exchange gases between the water and basically breathe through their skin. Frogs and other amphibians are now under attack by Cryptococcus. This fungus, with its flagellated spores, the spores will swim to the frog and block all of those pores in the frog's skin, making it impossible for the frog to breathe through its skin. When it can't breathe through its skin, it reduces its oxygen capacity, it gets really uncomfortable, it stops moving, and it eventually dies. So there's mass extinctions of amphibians going on right now due to this fungus. And there's also huge efforts to try to prevent this from spreading. So if chytrid fungus is found in a water source, they do their best to try to treat that water source in the safest way in order to save the amphibians. Select human pathogens. Dermatophytes are probably the largest category or most common category of human pathogens. Dermatophyte. Derma is talking about the skin, and phyte is talking about something that consumes it. So it's going to consume the skin or dissolve and digest the skin. These are fungal infections of the skin. Dermatophytes are not a genus and species name, so it's not a specific type of microorganism, rather it's a group of microorganisms. Some examples of dermatophytes include Epidermophyton flacosum and Microsporum gypsum. Trichophyton has various species as well, so those are the species, but dermatophytes, that's just a generalized category of fungi that infect the skin. So they cause infections such as ringworm, athlete's foot, cupitis, and others, and they are in the phylum Ascomycota. Dermatophyte regional infections, so Tinea cupitis, this is the scientific name for dandruff or fungal infection of the head. Tinea corporis is on the skin. Tinea cruris is in the groin, also known as jock itch. Tinea unguium is in the nails. And Tinea pedis is on the foot, so that's athlete's foot. So when we discuss the infectious cycle of this, signs and symptoms are usually redness, flaking of the skin. It usually looks pretty dry and doesn't have a very strong odor. If it does have an odor, it may smell yeasty or smell sweet. Transmission is usually direct contact. So you come in direct contact with the organism, and then the organism tends to overgrow at that time. Diagnosis. Dermatophyte infections can be diagnosed using skin scrapings. So you physically scrape part of the skin off onto an agar plate, and then you can allow it to grow. And once it grows, then you can evaluate the mold that grew on that plate. You can look at the color, shape, size of the colony, the texture of the colony, what the colony looks like on the top and on the bottom. And then you can also look at it microscopically. So you look at it under a microscope, you use that lactophenol cotton blue stain to safely stain it and look at it under the microscope. And you're going to evaluate it for the reproductive structures like the spores, and then the hyphae, and whether the hyphae are septate or aseptate, and what they look like in their arrangement and so forth. So that's how you diagnose them, usually by a skin scraping. If it is a toenail infection, you can implant the toenail directly into the agar plate, and then it'll grow from there. Treatment would be with just common antifungals. And prevention, it's really tough to prevent these. You have to know that it's present and avoid it. So avoid contact or direct contact with the fungus. So it's really tough to prevent this because oftentimes we don't see the direct contact. The next one I'm going to talk about is Candida albicans. Candida albicans is the causative agent of a yeast infection. Predominantly a vaginal yeast infection, but it can also cause oral thrush. And in more severe cases, in immunocompromised patients, it can cause pretty wicked meningitis if it gets inside of the cerebrospinal fluid. So different types of infections, but the most common is going to be a vaginal yeast infection. For signs and symptoms, there's going to be extreme itching of the vagina, and sometimes you will see a white discharge, and sometimes it looks clumpy. Some people compare this discharge to cottage cheese-like discharge, so it's really clumpy and nasty. The vagina itself is going to be inflamed and red and really itchy. Transmission, usually this yeast is commonly found on the body. It's found in your mouth, it's found in the vagina, it's found in different cavities in the body. The digestive tract has it, so it's naturally around. You get it from birth, you get it from the environment, you've naturally acquired this, but usually it's kept in check by your natural bacteria. When you take an antibiotic and you kill off some of your natural bacteria, or if you become immunocompromised or undergo a major surgery or something like that, then you give this opportunistic pathogen an opportunity to overgrow. And when this overgrows, it can cause severe infection. So it's usually an opportunistic pathogen that's already in your body, that something changes in your body and now it can overgrow. In the case of yeast infections, it's predominantly a pH imbalance of the vagina. So pH is very important. You need to have a lot of acid in the vagina to prevent the overgrowth of pathogens. If the pH of the vagina rises, for example, if you're using soaps inside the vagina, that's going to increase the pH. Semen is also going to increase the pH of the vagina. Those things can cause the pH to rise, which will cause an overgrowth of this stuff. The natural bacteria are there to produce acid, like lactobacillus produces lactic acid to decrease the pH of the vagina. But if that natural bacteria goes away, that's another reason why that pH could increase. One of the quickest treatments for an offset pH in the vagina is by taking ascorbic acid, which is vitamin C tablets. Vitamin C does have the potential to treat certain types of like bacterial vaginosis, but having a yeast infection is pretty severe. So just by simply adjusting the pH wouldn't be enough to treat the yeast infection. So treatment for this is going to be vitamin C tablets, and then you can take the antifungal as well. Diagnosis, usually it's just a simple swab. So take a swab of vaginal fluid, look at it under the microscope directly, and you'll be able to see yeast-shaped cells. Yeast are very large compared to bacterial cells, so you'll be able to see them clearly, and sometimes you'll be able to see the buds of the yeast. Other more specific ways to diagnose would be a serum tube test. So if you place yeast cells in serum, they grow this germ tube, and you can see this tube extending out. And that takes a matter of a couple hours. They'll grow this tube in human serum. So that's kind of an interesting way to diagnose. There's also molecular diagnostic techniques. You can culture it on a plate and grow it on a plate and look at it microscopically. So kind of a presumptive or a preliminary identification is just a vaginal swab, right from the vagina onto a slide, look at it and look for yeast cells. And a secondary causes, you would culture it and go from there. Prevention. It's really tough to prevent yeast infections from happening, especially since it's an opportunistic pathogen. So if you can try to stay healthy and try to avoid autoimmune diseases or fluctuations in vaginal pH, then you can kind of prevent it. But those things are difficult to prevent. So this is something that's already on your body that will overgrow when it has the chance. So it's really tough to prevent. Cryptococcus neoformans. This is another type of yeast. So Candida albicans causes the most common type of yeast infection. Cryptococcus neoformans causes another type of yeast infection, but this one's more common in cerebral spinal fluid. So it's a more common cause of meningitis, especially in infants. So for this one, signs and symptoms, you feel very ill. You have a high temperature, stiff neck, all of the symptoms that come along with having meningitis. Transmission, this one's transmitted directly by a infected individual. Diagnosis, so since this is found in a usually sterile site, such as the cerebral spinal fluid, you would take a CSF sample and you can grow or culture directly from the CSF sample, or you can look at the CSF sample microscopically to look for the presence of yeast. There's this special staining procedure called an India ink prep, and this is a picture of it right here. If you use India ink to stain the specimen, you'll be able to see a capsule around the yeast organism. So Cryptococcus neoformans is a capsulated yeast. So the organism's on the inside and it's surrounded by this gel-like capsule. It's one of the few capsulated yeasts that's going to be able to infect humans and be able to get in the CSF. So usually if you take a CSF sample, you put the India ink on it, and you see capsulated yeast, you can pretty much assume that it is Cryptococcus neoformans. There's also, of course, molecular diagnostics, and you can culture it on a plate and so forth. Culturing it on a plate is pretty dangerous. If you can diagnose it with just by looking at it here and you can treat it and the patient gets better, there's no need to grow large quantities of it in a lab and potentially expose other people to this. Treatment is by using antifungals, and prevention, it's tough to prevent. Usually it's going to be kind of acclimated to the more immunocompromised patients or those that are really young or really old and aren't able to fight it off. Prevention is just good hygiene and normal preventative measures for preventing any type of meningitis type infection.